Welcome to today's lecture on cell biology, specifically focusing on the intricate processes that occur during gut development and function. The human gut not only plays a vital role in nutrient absorption but also serves as a barrier against harmful pathogens. To understand the complexity of these mechanisms, we will explore various aspects including molecular mechanisms, cell differentiation, nutrient absorption, and immune responses. So let's dive right in. During embryonic development, the gut tube undergoes a remarkable process of elongation and folding to form the functional gastrointestinal tract. This process involves a precise orchestration of molecular events, including the regulation of cell proliferation, migration, and differentiation. Several key signaling pathways, such as the WNT, FGF, and BMP pathways, interact to control gut tube elongation. Additionally, the physical forces generated by the growing organs adjacent to the gut tube contribute to its folding. Understanding these molecular mechanisms is crucial for unraveling the etiology of congenital gut malformations and developing potential therapeutic interventions. The gut relies on the enteric nervous system, ENS, to control movement, secretion, and sensation. Neural crest cells, NCCs, are special cells that form the ENS, migrating from the neural tube to the gut. If NCC migration or differentiation goes wrong, it can lead to disorders like Hirschsprung's disease. Understanding the molecular signals that guide NCCs is crucial for ENS development and potential treatments. During gut development, the endoderm gives rise to various organs, including the liver, pancreas, and intestines. This process is tightly regulated by a network of signaling pathways and transcription factors. For example, the Notch signaling pathway plays a critical role in determining cell fate decisions within the endoderm. Transcription factors, such as SOX9, FOXA2, and GATA4, are essential for organ-specific differentiation and function. Dysregulation of these signaling pathways and transcription factors can lead to developmental anomalies and diseases, highlighting the importance of understanding their intricate mechanisms. The gut epithelium is composed of various cell types, including absorptive enterocytes, mucus-secreting goblet cells, hormone-producing enteroendocrine cells, and antimicrobial peptide-secreting Paneth cells. The differentiation and maintenance of these specialized cell types are tightly regulated at the transcriptional level. Key transcription factors, such as MAth1, Phe1, and Hes1, drive cell fate decisions within the gut epithelium. Understanding the regulatory networks controlling gut epithelial cell differentiation will shed light on how intestinal homeostasis is maintained and how dysregulation can lead to gastrointestinal diseases. The gut epithelium has the ability to regenerate and repair itself, thanks to intestinal stem cells, ISCs, found in specific areas called crypts. ISCs give rise to amplifying cells that then differentiate into different gut cell types. Recent techniques like lineage tracing and single-cell RNA sequencing have enabled the identification and characterization of different ISC groups in different gut regions. Understanding the self-renewal and differentiation mechanisms of ISCs could have significant implications for regenerative medicine and gastrointestinal disorder treatments. Microvilli are finger-like protrusions that significantly increase the surface area of absorptive cells in the intestine, enhancing nutrient absorption efficiency. These microvilli are rich in actin filaments, membrane-associated proteins, and transporters. The brush border enzymes located on the microvilli digest carbohydrates, proteins, and lipids, facilitating their absorption. Dysfunctional microvilli, as seen in microvillus inclusion disease, impair nutrient absorption and result in severe malnutrition. Understanding the ultrastructure and molecular mechanisms underlying microvilli formation and function is crucial for deciphering how absorption occurs and developing novel therapeutic strategies. The intestinal epithelium acts as a protective barrier, allowing nutrient absorption while blocking harmful substances. Tight junction proteins like claudins and occludins maintain this barrier. Mucus-secreting goblet cells also contribute to protection. Disruptions in these proteins or mucus production can cause inflammatory bowel diseases and gastrointestinal disorders. Understanding these mechanisms is crucial for gut health. Carbohydrates are important for energy and need to be digested and absorbed. Salivary and pancreatic amylase break them down. Specialized transporters like SGLT1 and GLUT2 help move glucose and fructose across the intestine. Problems with digestion can cause malabsorption syndromes, like lactose intolerance. This knowledge helps us understand nutrition and digestive disorders. Proteins are essential for growth, tissue repair, and numerous physiological processes. In the intestine, dietary proteins are broken down into amino acids by proteolytic enzymes, such as pepsin in the stomach and trypsin/chymotrypsin in the small intestine. The resulting amino acids are then transported across the intestinal epithelium by specific transporters, such as the SLC7A9 protein transporter family. Disorders in protein digestion or amino acid absorption can lead to various nutritional deficiencies. Understanding the enzymatic processes and transporters involved in protein absorption provides insights into disorders such as celiac disease and protein malnutrition. In the video about gut development and intestinal absorption, we discussed how the body breaks down and absorbs dietary fats. Bile salts from the liver break down fats, which are then absorbed by cells in the small intestine. This process is important for energy and cell function. Problems with this process can lead to malabsorption disorders. Studying these processes helps us develop treatments for lipid-related disorders. Enterocytes lining the intestinal epithelium play a vital role in nutrient absorption. Nutrients such as sugars, amino acids, and electrolytes are transported across both the apical and basolateral membranes of enterocytes. These transport processes are carried out by specific membrane proteins, such as SGLTs and GLUTs for sugars, and SLC transporters for amino acids. The regulation of nutrient transport is influenced by various factors, including hormonal signals and luminal nutrient concentrations. Understanding the intricate mechanisms and regulatory cues involved in nutrient transport provides insights into nutrient absorption disorders and facilitates the development of therapeutic strategies. Gut-associated lymphoid tissue, GALT, is a part of the immune system protecting the intestine. It consists of lymphocytes, dendritic cells, and specialized epithelium. GALT fights pathogens and maintains immune tolerance. Dysregulation leads to inflammation, autoimmune diseases, and infections. Studying GALT helps treat gastrointestinal immune disorders. In summary, the gut undergoes intricate development and functions through various cellular and molecular mechanisms. Understanding these processes is crucial for understanding gut function and potential interventions in gastroenterology.